Second part, I want to move on to this preclinical mouse model. So this was our first attempt to make a serious analysis of and use the power of the Evosep and try and analyze a few hundred samples. So one model that has been used now extensively in our lab, this is primarily work from Evan Williams and Yibo Wu in the lab, in the Eversol lab, is this BXD mouse collection. So this is currently the largest and best characterized mouse genetic reference population. So essentially what they do is they take two laboratory mouse strains, this C57 and DBA strains, and they do a lot of complicated breeding, which I frankly don't understand, but they end up with a very well-defined set of 183 mouse strains, which are very well genomically characterized, and these mice can then be used for genetic association studies where you would try to associate, for example, phenotypic features of these mice with particular genetic features, or indeed molecular features, so protein sort of QTL analysis. Can we associate the abundance of a protein or a transcript or a metabolite with a particular genetic locus? And so Evan is continuing to work on these, and the latest study he's been working on for the last few years is a very large-scale study where they try to use this BXD mouse population to look at molecular physiology across tissue, time, diet, and genotype. So they take a whole bunch of mice from these strains, they feed half of them a chow diet, half of them a high-fat diet, and then they allow some of these mice to die with a natural lifespan. They also sacrifice a large portion of these mice and collect tissues at different stages along the way. And then they're doing a kind of a multi-omics approach here, measuring genomes, transcripts, proteomes, and eventually metabolomes. And so we thought that this would be an interesting test case for the Evosep method. So we had already acquired a bunch of data on roughly 400 of these mice using a kind of our more standard workflow on the 6600 with the exigent nano-LC using a 60-minute nano-LC gradient. And we decided to take the same set of samples and essentially inject these using the Evosep methods with a 21-minute method using this, which allows you to analyze, it's a 21-minute gradient, it's a 24-minute method, but it allows you to analyze 60 samples per day. And you can see some of these comments which came from Evan. So it took essentially, whereas the numbers basically around 8 weeks to acquire the data in this way. And you'll notice that that's not, the number of samples multiplied by the runtime, but there's also things that happen in the middle where there's some downtime. You have things like, you need to occasionally clean the machine, you need to tune it occasionally, you need to change the LC column, and occasionally you get some auto-sampler failures that don't really work properly. This also, the Evosep is not entirely free of these effects, but it seems like that they're reduced to some significant degree. So this data acquisition, which took us about 8 weeks in our kind of more standard workflow, we were able to do in a little bit more than a week using the Evosep platform. And essentially, we didn't actually clean the machine until the end. And these are liver samples, so they're not the cleanest samples necessarily in the world. But we were able to get all 400 samples through without having to clean the instrument. We didn't have to do any LC column changes. So this is already a good start in our view. Okay, so let's look at a little bit of the data from these mice. So the one thing is retention time stability, and we always are spiking in these IRT peptides for doing retention time alignment. And one thing it's easy to do is to take these IRT peptides and say, are they stable in retention time space across time, right? So this is two of these IRT peptides on the Evosep. These are two of the same two IRT peptides in our nano-LC 60-minute gradient, and I think you can appreciate that the stability in the retention time domain is actually very nice, and I think that these kind of jumps here are also LC column changes. So this already looks pretty good. What about quantitative stability? So Evan plotted here the Spearman correlation for some different kinds of replicates. So if you have an adjacent technical replicate, these are basically the protein abundances for a few thousand proteins. So this is run 79 and run 80, which are adjacent technical replicates, and we get very good correlation, Spearman correlation on the order of 0.97. If we look at distant technical replicates, run 79 and run 406, then we're getting still very good correlation, not quite as good as the adjacent technical replicates, but still fairly impressive. And again, you can look at adjacent different samples and distant different samples, and the correlations are a bit lower, but these are different biological samples. So this actually looks quite nice to us, and I think it's actually, I'm not showing the numbers, but it's very comparable to what we saw in the longer gradient data set. So this is also seems like a good indication. What about the numbers of peptides and inferred proteins that we can detect? So on the Evosep, we had essentially 2,800 proteins using proteotypic peptides. On the longer gradient, that was more like 3,000. So that number is not very different. The peptide number is different, right? So we go from 18,000 to 32,000, and there's a relatively large overlap in these. And you can see that the numbers of peptides are fairly consistently detected. Either in the Evosep or the exigent method, and there are some things drop out here, but we can usually assign this to some technical artifact, either related to the sample prep or potentially to some other technical issues. But in general, we think this is very, very consistent. So the message here is, yes, we have some losses at the peptide level, but we think that at the protein level, we're doing reasonably well. And so just one slide to show that some kind of known biology is recovered using this data set. So here is, so Evan knows these mice very well, and there's a lot of kind of true positive things that he knows should be there. So for example, this enzyme DHTKD1, he knows that in red we have the allele from this DBA mouse, in blue we have the allele from the C57, and he knows that there's a genetic difference here, essentially, that should change the abundance of this particular enzyme. And what you can see is that on either methodology, we can recover this difference very well. And the gray in the middle is the heterozygous mice, so you kind of expect those to be somewhere in the middle. And Evan has a whole bunch of these, not just this one. I'm just showing one example, but he was very satisfied that his ability to recall known biological information from the Evosep data or from the longer gradient data was roughly equivalent. To the extent that I mean, all of these mouse studies that he's now continuing to do, he goes directly to the fast way of doing things, right? So it's really kind of made a big difference. So the conclusion from the second part is that we have this preclinical mouse model with, I mean, there's really a dramatic increase in throughput, and not just the kind of theoretical time that it takes to do the measurement by multiplying the number of samples times the number of ones, but I would say the kind of real, well, what I would call the real instrument time, right, accounting for kind of downtime and maintenance, we seem to do much better there. There's a sort of modest loss at the protein level, modest loss of information, but I think it's one we think we can tolerate in order to have this really dramatic increase in throughput. I didn't show data on this actually, but it does seem like there's lesser batch effects. So in these, 400, if you inject 400 samples, you usually have to do a lot of normalization and a lot of batch correction in order to make the data look actually reasonable. And it does seem like the need for that is reduced with the Evosep data, because we're acquiring the data so fast and in a relatively uniform way. So we still need to do normalization. We still need to do some batch effect correction, but it seems like it can be a bit less aggressive than what it was with the longer gradient data, and of course if you can do less of this batch correction and normalization, that will benefit your data in the end. And so, as I said, Evan is doing more large-scale analyses with hundreds of mice. I mean, a couple of months ago he said, I want to analyze the brains of a couple of hundred mice, can we do it? And it was just a question of doing the experiment. It took much less thought and planning than it did to do the kind of longer way of doing things. So I think it's actually changing a little bit the way that we think about what we can do, having access to such a fast technology. And the quote from Evan is that there's no way he's going back to the old way of doing things. So that's this mouse study.